Today's podcast is going to focus on drugs that have been in the news lately for its beneficial effects in obesity and previously used in diabetes. And this drug and others are in a class called GLP-1 receptor agonists. And so my guest today is Professor Tom Foltynie. He's a physician scientist at the University College London Queen's Square Institute of Neurology. His has training in epidemiology, some genetics and biostatistics, and he's running a lot of clinical trials. He's done a lot of trials and I guess troubleshooting and deep brain stimulation for Parkinson's disease. His main focus is movement disorders, Parkinson's disease being the main one. But more recently, he's done some exciting clinical trials with these GLP-1 receptor agonists in Parkinson's patients. So welcome, Tom.
Thank you very much, Mark. It's a pleasure to be here and to chat with you.
And you did your MD and PhD at Cambridge, and of course that's where you got interested in what you're doing now. And there you were with Roger Barker, who's very well known in the neurodegenerative disorder field, and you were interested initially in the genetics, right?
Yeah. So Roger said to me, well, we need to cure Parkinson's with cells, but the trouble is, Parkinson's patients seem to be different. So, we need to just work out about the different types of Parkinson's. So can you just, do a study and collect patients and then put them into groups and understand the heterogeneity? Now, that sort of idea didn't stand up to a great deal of scrutiny when you try and do it with proper epidemiological methods. So the Wellcome Trust, they said to me, you need to do some proper epidemiology training. And we ended up doing all of the biostatistics and epidemiology courses in Cambridge. And that led to us setting up this incident cohort. We recruited a population-based cohort of patients with Parkinson's disease from diagnosis. And we did all the clinical measures, and we collected blood samples, and we did genotyping. We did the first genome-wide association study in Parkinson's patients, hugely underpowered. We didn't have enough to identify the risk genes, but we learned a lot about some polymorphisms and how they may influence some of the cognitive features of Parkinson's. So at the end of all that, I said to Roger, well, it's not so easy. You can cut the cake in lots of different ways. You can talk about the motor phenotype. You can talk about the genes. What we need are better biomarkers, which we'll understand today. So that was a long time ago.
Yeah. And was Andy Singleton over there at that time?
He wasn't with me in Cambridge at the time. Of course, I know him from subsequently. He's been leading all the genome-wide, up to the latest GP2 work in genetics and Parkinson's now.
Yeah, he's at NIA where I was, another lab chief. Okay, so then you got in as far as clinical trials. Your first work, you did a lot of work on deep brain stimulation, which it's used in practice in some patients. Can you talk a little bit about that? I had a previous podcast on Parkinson's with Ted Dawson, and we just had him go over kind of the clinical presentation, how do you diagnose it, and what's kind of the cellular current understanding of the cellular molecular. We didn't really get into treatment at all. So can you say, I guess, kind of describe the different parts of the brain controlling motor function and what you do, where you put the electrodes and why you put them there?
Sure. So, when we first see people with Parkinson's, unless there are red flags that there may be something else going on, you can look at tremor, rigidity, bradykinesia, and you can be fairly confident that if you replace their dopaminergic deficits with standard drugs, levodopa and the dopamine agonists, then patients will come back the next visit and be very grateful. They'll feel there's been a weight lifted off their shoulders. They'll be walking and turning more easily, with better dexterity. And so they'll go through a honeymoon period where their dopamine symptoms are to a greater or lesser extent relieved by the levodopa, and it works well. But that honeymoon period, is a variable duration, and within a few years, people will start developing motor fluctuations. That's where the drugs will work, but then they'll wear off after a period of hours, sometimes unpredictably, and then they have these on-off fluctuations throughout the day where they can be good and then disabled, then good and disabled again. And you increase the dose of levodopa replacement, you add accessory drugs, and you can get to the point where people develop involuntary movements such as dyskinesias. So these are, writhing movements which can be just as disabling as the poverty of movement of Parkinson's disease. And so going between these off periods, these dyskinetic periods, can be very difficult. And so at that stage where we've, in a sense, exhausted the easy drug therapies, we move on to what we call the advanced therapies. And the most popular of these is deep brain stimulation. This is a tried and tested way of intervening. It's been around for 30 years now. So it involves putting a wire, one each side of the brain, deep into the brain. And there are different targets that can be manipulated, but the most common target is the subthalamic nucleus. And if you put where a neurosurgeon puts a wire precisely into the subthalamic nucleus, you can record from the cells there, and you find that they're misbehaving. Rather than having beautiful neuronal firing patterns which facilitate movement, they start oscillating in a synchronized manner. So, and they tend to all fire together in the beta frequency. So this is the frequency between 12 and 30 hertz, so 12 and 30 times a second. And this beta frequency seems to correlate very tightly to the slowness and stiffness of Parkinson's disease. So if you put these wires in place, you connect them up under the skin to a pacemaker, then you can turn on tiny amounts of electricity and deliver electricity which can obliterate these beta oscillations, and it can allow more normal firing to resume, and it tends to get rid of the slowness and stiffness of Parkinson's disease in a very satisfying switch on and offable way. So it's a very effective treatment. It's certainly not a cure for Parkinson's. If you turn it off, all the symptoms come back, but it can be a way of reducing the severity of the off periods of Parkinson's. And by doing that, you allow people to reduce their medications, so that can get rid of some of the dyskinetic side effects or other side effects that they may have developed because of their medications. So it's a very effective treatment.
So this is essentially bypassing the dopaminergic neurons?
Yeah, it's dealing with the consequence of having a low dopaminergic state.
Yeah. All right, and then, in the early stages of Parkinson's disease, then there's clearly a big need for some treatment that can slow the progression. Like all these diseases, you want early diagnose it as early as possible, and start to treat it before it gets really bad. So how did you get interested in the GLP-1 receptor agonist, exenatide or exenin-4?
Well, I suppose it's inevitable when we look after people with Parkinson's at all stages. You look after people with DBS after 10 or 20 years. The DBS may be helping slowness, stiffness, and tremor, but they're no longer the problems. Their problems are speech, they're unintelligible speech. They have balance problems. They're falling over. They have cognitive problems. They have hallucinations and psychosis and depression, and so it's no longer a dopaminergic movement disorder. So realizing that we're in desperate need of something to either hold people in those early years where they have a treatable motor disorder, or even better, prevent them developing that in the first place. This is clearly what we need to do. We've got an aging population, so this is getting a bigger problem. This isn't going away. So I was in this setting, doing DBS related research, but having had an education on epidemiology and biostatistics, and that included a lot of trials methodology, where I got in touch with the Cure Parkinson's Trust and a chap called Richard Wise, who was the director of research. he said to me, "We're very interested in this drug called exenatide, and we want you to set up a trial." Which was we'd done some trial related work in DBS. I had an education in trial methodology, so I thought, yeah, why not? But this was in quite a naive space, if you like. I hadn't done a disease modifying trial in Parkinson's disease, and we didn't have a big commercial backer. So we looked to see, how much it would cost to get access to drug and placebo. And the drug we could buy off the shelf, but the placebo wasn't available from the manufacturer. So for us to make it was going to cost millions. And so we ended up contriving that we would do a small open label trial. We'd randomize patients into two groups, and they'd all have their standard treatment, but half of them would have the exenatide injections and the other half wouldn't. So everyone would know which treatment arm they were in. And we were aware that, of course, we'd be criticized. Well, what about placebo effects and observer bias and all that? But we did our best to mitigate that by videoing the patients at the start and videoing them periodically throughout the trial. And then someone who is independent from the trial team could rate the severity of the Parkinson's disease. So it became single-blinded.
Yeah.
But of course, there's still the potential placebo effects that the patients can experience. So that's how we conducted the first trial. This was using Byetta, which is the twice daily injection. So it's a lot more intrusive to patients than the later version by Durion, which we used in our second trial.
Yeah. And so the first trial is a small trial. It wasn't double blind. And how long were they treated, and what results did you get?
We kept it going for a year, and I'm hugely impressed with the sort of the ability of Parkinson's patients to stick to a protocol.
Twice.
Parkinson's patients are fabulous for trials. They're slightly introspective, and they want to be meticulous about how they are and how they fill in the questionnaires. And we had people that were developing their own outcome measures in addition to what we were asking them to do. And all of the patients, it was only 44 people, all of them completed the whole 12 months of trial injections and trial visits. And we decided that having identified that there was an advantage in terms of movement, we use the UPDRS Part 3 as the movement scale to measure Parkinson's, but they also had an advantage in terms of their cognition. So we used the Mattis Dementia Rating Scale and showed that people on exenatide were better at the end of 1 year than people who were acting as controls. So we decided at that point, why don't we just keep following these people up for another year, even if they're not using the injections anymore, and just see what happens in the longer term. So at the end of 2 years, patients who had exenatide were still better off in terms of their movement and their cognition than people who had been randomized to be controls. And so that, in a sense, added a little bit of extra credibility to this small open label trial. And it kind of de-risked future investment, because moving on to do a double-blind placebo-controlled trial was a much more expensive undertaking. And so we needed bigger funding.
Yeah.
And that meant that funders had to be a little bit more confident that there was going to be the potential for a return on that. So that's how it went.
Yeah. And then, so that was like about 10 years ago. And then more recently, you have done a randomized controlled trial and had a paper in the Lancet reporting those findings. Can you talk about that?
So based on the first trial, we had a nice lunch with the Michael J. Fox Foundation, and they said, they're very interested in this. We costed up what it would be for a double-blind placebo-controlled trial. And the other important issue that we managed to get the manufacturer of the drug also bought into this. So at the time it was Bristol Myers Squibb, and that they said, we'll give you drug and placebo for 60 people. So they stipulated that the size of the trial. This is how much you can get. And then we worked backwards from there to calculate how much the trial would cost. And the Michael J. Fox Foundation said, yep, they'll be willing to foot the bill for that. So again, this was a 1-year treatment exposure. We did the same types of scales using the UPDRS Part 3 to measure movement problems, doing cognitive scales, getting blood samples as well, and we did lumbar puncture collection as well, so we could try and learn a little bit more about Parkinson's disease in parallel. And again, now in a double-blind placebo-controlled trial, we show that there was a clear difference between those people with exenatide and those people on placebo. So often there's concerns about the placebo effect and people getting better because they believe in a drug. But in our trial, those patients on placebo just had a steady deterioration over the whole of the 48 weeks that they were injecting placebo. And there was no placebo effect at all. I did wonder if this is because of the measure we'd chosen, which is this UPDRS Part 3, but we did it first thing in the morning before they'd taken any of their dopaminergic drugs. It's likely because we did that, then people can't, in a sense, express the placebo effect. They haven't got dopamine. I mean, a lot of placebo is mediated by dopamine in the brain. And so we using this measure, we didn't get a placebo effect. What we did find is that those people using exenatide had an early improvement in their symptoms, and then it remained fairly static. There was no decline over the next 48 weeks. And then they had a small dip at the end of the 48 weeks. We had a 12-week washout period. But looking at the two arms, that there was clear daylight between the two groups, about 4-point difference in the UPDRS Part 3 between placebo and the active treatment arm. And that's when the drug is completely washed out. So we're hoping that would eliminate any possibility for there being a symptomatic effect, and this actually was more evidence for disease modification. But we're still, discussing internally and with colleagues, how is this working? Is it symptomatic? Is it disease modifying? If it's disease modifying, how does it work? And how do we better understand this? And so we've had a lot of thoughts about mechanism and how that translates to the clinical.
Yeah, and we can get into that pretty quickly here. So the Lancet trial, was that the twice daily or the once weekly?
That was a once weekly. So
once weekly.
Yeah.
Yeah, that's really as far as a patient standpoint, that's just a fantastic thing. Once weekly.
Yeah, and again, the compliance with the trial was amazing. That we had basically one or two missing data points from the entire cohort of people for the entire year. There was no dropout, and there were just 1 or 2 missed visits because of inconvenience that coincided with things I had to do. So it was hugely powerful, and of course that meant that we had enough ammunition to convince the government funding in the UK to fund a phase 3 trial of using Bydureon, which is what we're doing at the moment. And we'll finish, the last patient will have their last visit in February 2024. So
and Tom, is that a multi-center trial or?
Yes, yeah. So that's 6 sites in the UK.
Yeah.
So ourselves, Plymouth, Oxford, Manchester, and Edinburgh, and we'll follow patients up for 2 years. The idea being that if there's a difference at the end of 2 years, then it's great, but that won't answer is it symptomatic or disease modifying. But we'll have data along the way, and if we find that there's divergence between the two arms, then that's clearly above and beyond a symptomatic effect. It's doing something for the rate of decline, and so that's in a sense what the new trial will give us, as well as being multi-site and more patients. It'll give us that additional information.
Okay, let's go to the science now. So work backwards. All right, so GLP-1, glucagon-like peptide 1, what is it? So,
there's a great story in this, and I'm sure you know it very well.
Yeah.
A chap called John Eng was torturing Gila monsters in the Arizona desert one day, and looking to see what he could find in the saliva and venom of the Gila monster. So, people may know that this is a slightly unusual lizard that only eats a couple of times a year, so it has to have a very close control of its own metabolism. And this isn't a bizarre behavior. This is a way that many drugs have been discovered, or many important proteins have been discovered. I tell people that heparin, which we use as an anticoagulant, was found in the venom of the Malayan pit viper. And so this is not an unusual way of making new discoveries. So John Eng found this protein exendin-4, which is 50% homologous to human GLP-1. So glucagon-like peptide 1 we all release as a hormone from the cells of the intestine in response to a meal, and it circulates in the bloodstream and has an effect on GLP-1 receptors in the pancreas. It only works for a few minutes because it's broken down by the enzyme DPP4. But exendin-4 and the synthetic version exenatide doesn't get broken down by DPP4, so has effects in the bloodstream for many hours. And what we've learned is that there are these GLP-1 receptors not just in the beta islet cells of the pancreas, but they're in many tissues throughout the body. And so many investigators are interested, well, what happens when you stimulate GLP-1 receptors in the liver, the heart, the fat, or in our circumstance, the brain?
And so GLP-1 increases insulin release from the pancreas. There's also evidence it increases insulin sensitivity, that is the ability of your cells, muscle, liver, and so on to remove glucose from the blood. So that's good for diabetes. And another thing it does, I guess you probably saw this in your clinical trials, it inhibits appetite. It can suppress appetite somewhat, so people may lose a little bit of weight. Yeah,
definitely. And of course, that's great if people are overweight. But if people are underweight or struggling with their nutrition, having this sensation of bloating or anorexia is an unwelcome and unwanted side effect. It tends to be fairly transient. It's the side effects from exenatide tend to last for the first 3 months or so, and whether it's delayed stomach emptying or nausea, those types of things, they tend to last for about 3 months, and then they tend to settle. So hopefully, if you can get people through those early weeks, then they can remain on the drug.
Yeah, and I think so. I was recruited as a lab chief to NIA and started in the year 2000, and that was the time. So for diabetes, actually, Josephine Egan, who's a diabetes expert at NIA, developed it for diabetes, did clinical trials in, I think it was the clinical trials were in 2002. But then Nigel Gregg, who was a senior investigator under me in my laboratory, when I came there, we'd done a lot of work on cell culture and animal models that are relevant to Alzheimer's, Parkinson's, stroke, et cetera, and we were culturing primary neurons from brains, mainly from the hippocampus. So so one morning a week, I'd spend the whole morning dissecting brains of embryonic rats, so up to 50 embryos, dissect out the hippocampus, collect them all, then dissociate the cells and put them in culture. And they grow axons, dendrites, form connections. And we've done a lot of work on something called excitotoxicity, which is neurons. Glutamate's the main neurotransmitter in the brain, and it's critical for all our behaviors. And dopamine, the only way it affects behaviors is really by modifying activity in glutamatergic neurons. But anyway, in epilepsy, you get severe activity, neurons can be damaged and killed by a process called excitotoxicity. I think that in Alzheimer's, Parkinson's disease, there's an excitotoxic component to the neuronal degeneration. So anyway, having said all that, some of the first experiments Nigel and we did was just take cultured neurons, treat them with GLP-1 itself, or exenin-4, this Gila monster 39 amino acid peptide, and look, see, are they more or less vulnerable to being damaged by glutamate? And we found they were protected. And then Nigel did a lot of work. We helped him with some in vivo models of Alzheimer's, Parkinson's, and actually, it was, I think, that the PNAS study that from mainly Nigel's lab, but with help from other labs, in a Parkinson's model. But that kind of also was involved in prompting, I think, the clinical work. So anyway, as you mentioned,
that's the unique thing about exenin-4, exenatide, is that it seems to have these positive effects, not just in one model or one cell system. It's whether it's, you know, for Parkinson's, 6-hydroxydopamine and MPTP and lipopolysaccharide and excitotoxicity. And more recently, there's Ted Dawson's alpha-synuclein transgenic animals, as well as looking at the cell cultures. So it seems to have these neuroprotective effects, not just in one lab, in one model. It's across labs and across multiple models. So it's
yeah. And my impression is, you know, and earlier on you were talking about, well, is this really modifying the disease process in Parkinson's? That is the actual degeneration of the neurons, or is it just somehow enhancing the function of dopaminergic neurons that are still there, or maybe doing other things to existing neural circuits? But this kind of broad spectrum of effects would seem to lean to point to disease modifying, and just in my view. Now, we can get into the receptors. Do you want to talk a little bit about the receptor itself for GLP-1?
Well, we can talk about insulin resistance. I'm sure we will, because I suspect that this is the key to what is happening with, you know, downstream from GLP-1 receptor stimulation. You know, I'm no expert about, you know, precisely how the GLP-1 receptor.
I can help out there. But talk about the insulin resistance, and you followed up, you know, Dimitrios Kapagiannis, and again, he was a tenure track investigator in my laboratory, and you know, he did. Let's talk about this extracellular vesicle study, because this gets to the insulin resistance thing. So.
Absolutely. So, you know, we're basically piggybacking on what's been learned in the Alzheimer's world.
Yeah.
And it appears to be very relevant to Parkinson's disease as well. And it may be relevant to other forms of atypical Parkinson's. So we can come back to this, but we're also, you know, very interested in multiple system atrophy and possibly also in progressive supranuclear palsy, which are all forms of atypical Parkinson's. But it's increasingly interesting what this phenomenon of central insulin resistance, rather than peripheral insulin resistance we see in diabetes. This phenomenon, which is not so much involved in glucose uptake, but is involved in cell survival. So insulin signaling, of course, as you know, has an impact on the IRS1, this insulin receptor substrate. And a neuron, or perhaps even glial cells, can be insulin resistant or sensitive, depends on the phosphorylation status of IRS1. And if you, in an intact insulin signaling pathway, all the downstream pathways, including AKT and its phosphorylation and mTOR and its phosphorylation, those seem to have big effects on things which everybody accepts are important for Parkinson's. So that's alpha-synuclein oligomerization, it's mitochondrial dysfunction, and neuroinflammation. So all of those can be tipped in a negative direction as a result of your insulin path.
And impaired autophagy as well.
Yes, yeah, absolutely.
Which fits in with the mTOR.
Yes.
Yeah.
So when we talk to most people about Parkinson's pathophysiology, everyone's very comfortable talking about synuclein and neuroinflammation and autophagy and mitochondria, but very few people are yet focused on insulin resistance. Whereas in the Alzheimer's field, a few years more established, that there's a lot more work and credibility about this insulin signaling pathway being very, very relevant for neurodegeneration. So I think we're dragging our Parkinson's colleagues along to catch up with the Alzheimer's field, and I think we're now on the back of the phase 2 trial that we completed. We're in a much stronger position. So the blood tests that we collected during the course of that trial, with Dimitrios in NIA and Dylan Athauda, who works with me, from these blood tests, they were able to separate out extracellular vesicles or exosomes, and then they could, from them, separate those which were a brain neural cell origin. This is using the L1 CAM surface marker. And once they have their population of L1 CAM exosomes, they can then see what they contain and how that changes longitudinally through the duration of the trial, and if that differs according to exenatide or placebo. And what we found is that the patients, at the start had a very similar level of insulin resistance based on this IRS1 phosphorylation status. But over the course of the trial, the tyrosine phosphorylation, which makes people more sensitive to insulin signaling, that increased in the exenatide treated group, whereas it had a gradual slow decline in the placebo treated group. At the end of the 48 weeks, the tyrosine phosphorylation dipped again. So it does seem like you probably need to have ongoing GLP-1 receptor stimulation to maintain your insulin signaling pathway. But while that's nothing to do with hiding Parkinson's symptoms, it's enabling cells to function, and therefore make dopamine and have synaptic communications with other cells downstream. So this whole concept of whether something's symptomatic or disease modifying needs to be shaken up a little bit by understanding what mechanisms we're engaging with. So this is a really, really rich area of study.
Yeah, and the GLP-1 receptors coupled to adenylyl cyclase and cyclic AMP, which is an intracellular messenger, and also calcium, increase in calcium levels. And it's interesting that pathway, and I think that one of Nigel's, can't remember her name, did some experiments in the cultured neurons and showed that
Tracy Ann Perry.
No, it wasn't Tracy Ann. It wasn't a Brit. It was a Chinese. What's her name?
Lee, Yazoo Lee.
Yeah, that's it. Yeah. She was the first author on the Parkinson animal model. But anyway, bottom line is she found that GLP-1 and exenin-4 will stimulate BDNF production, brain-derived neurotrophic factor. That could play an important role because it supports neural survival, and there's actually a lot of literature on that, right?
Yeah.
So that's another way. So in addition to increasing the insulin sensitivity of the neurons, which your data suggests that even the neurons in the brains of these Parkinson's patients may increase BDNF levels. Yeah. And there was, were there trials of BDNF in Parkinson's? I can't remember.
So there've been a lot of trials in GDNF, so glial cell
that's
neurotrophic factor. People have been interested in BDNF and its impact on the tract B receptor. More because there's a functional polymorphism in BDNF, which is perhaps relevant in terms of not necessarily risk for developing Parkinson's, but some of the phenotype. We published a paper on that BDNF polymorphisms influence the risk of dyskinesia, early onset of dyskinesia, which is perhaps an important synaptic problem.
Is this the same polymorphism that's associated with depression?
I think it is, yes. Yeah.
Interesting. And in the same way, if you look, so with Ted Dawson, we did talk about the story of, Parkinson starts in the gut and goes to the brain. So there are these non-motor symptoms of Parkinson's. For example, my understanding is that, early on when the people were studying Parkinson's, they looked back in the clinical records, and the patients would often have reported like chronic constipation.
Yeah.
Something like their gut motility is impaired, or there's some problem with gut motility. And then, anyway, we don't need to go into detail on the whole story, but there's pretty compelling evidence that maybe this alpha-synuclein protein that's accumulating in neurons, it's first occurring in the gut and neurons innervating the gut. So did you look at any non-motor symptoms in the clinical trials at all?
Yeah, we did. I mean, there's standard questionnaires. There's the non-motor symptom severity scale, or even in the UPDRS, part 1 is looking at non-motor symptoms. Overall, we didn't find that there's a big impact. We looked in a post hoc way specifically at depression, because when you look at the whole scale, it's covering everything. It's covering bladder, bowel, blood pressure issues, sweating, saliva. And so if you don't have an effect on everything in the same direction, of course, the whole scale is not going to shift. But if you look at sub items, we did find that patients on exenatide had better scores for depression, which of course is very important in people's quality of life and well-being.
The BDNF is going up.
Yeah.
Maybe.
And also apathy, which is another major issue in Parkinson's disease. We didn't find any changes on constipation, but of course, one of the side effects of exenatide is it can slow gastric emptying. And so, irrespective of any central effects, there may be peripheral effects which confound the GI symptoms. So I wasn't surprised there. But you're absolutely right. It's Parkinson's shouldn't be viewed as just a motor disorder, slowness, stiffness, and tremor. It's very much a whole body disorder. It may well start in the gut for some people because they've ingested something which leads to alpha-synuclein misfolding in the gut. For other people, they may have inhaled something, and that's why they get olfactory deficits, as a first symptom, because they've got pathology in the nasal mucosa. And for yet others, it may be an inevitability that they've just got too much alpha-synuclein in the brain because of polymorphisms or mutations in their synuclein gene or their lysosomal genes. So it's a heterogeneous group. Back to Roger Barker's question.
Now, these GLP-1 receptor agonists have been used for type 2 diabetes now for at least a decade, I think. And is that enough to get any epidemiological data on whether diabetes patients are more or less likely to get?
Absolutely. So we've published a nice paper a couple of years ago on this. So diabetes is definitely a risk factor for Parkinson's, and it's a dose-dependent thing. The earlier your diabetes, the bigger your risk. So it can be up to a 4-fold increased risk of developing Parkinson's if you've got young onset diabetes. On top of that, diabetes makes your Parkinson's worse. Your rate of progression for both movement and cognitive problems, both of those things accelerate more quickly if you have diabetes. And it's not just because of small vessel disease or peripheral neuropathy that can occur with diabetes. It seems to be independent of those things. So we, on the basis that the GLP-1 receptor agonists have been around for a while now, we looked at the THIN database, which is a UK-wide GP health records. And we looked at every patient who had an instant diagnosis of diabetes. So we knew every drug that they've been treated for their diabetes from day one, and looked to see of those patients what the risk factor developing Parkinson's was, which is elevated, as we already knew, and whether that risk varied according to the drugs that they've been given for their diabetes. And so we found that overall, let's say your risk of developing Parkinson's in diabetes patients, your risk is, let's say, 10 per 100,000 person years. If you're treated with a glitazone, it is slightly reduced, so you have 8 per 100,000 person years. If you're treated with a DPP-4 inhibitor, your risk is 5 per 100,000 person years, whereas a GLP-1, it goes down to 4 per 100,000 person years. So it's having
50%.
have less than half the risk
Yeah,
of other people with other treatments.
that's dramatic. So what, besides drugs, we know that exercise, regular exercise, improves insulin sensitivity.
Yeah.
And there's some evidence that regular exercise during adult life reduces risk for Parkinson's, I think. We did a lot of what you mentioned, Cure Parkinson's Trust or whatever it is.
Yeah.
Earlier on, I got invited to go to a couple of their meetings in Michigan. They have this like yearly meeting where they kind of and prioritize, what I think needs funding. And so I pitched intermittent fasting, right? Because we'd done all this work in my lab on intermittent fasting, which improves insulin sensitivity, and it was beneficial in some Parkinson's models. So I think, people listening to this, we're all, my view is you, me, everybody, as we get into our 60s and 70s, we're all at risk of developing Alzheimer's or Parkinson's. So the more we can do to keep healthy lifestyle, the better. And, maybe delay a little further.
Absolutely. I suppose that the problem is that it's the most educated who are likely to be those who do the best things for their health, and they will exercise more. They may have gym memberships that other people can't afford. They may have the best diet, and they avail themselves of the best medical literature, and so there'll be early adopters of GLP-1 receptor agonists, as and when we can demonstrate that they're effective. I suppose what we need to find is, on a population basis, measures that can help the less educated, the less well off, and make a difference. And you have to make it as easy as possible. If for some people, changing diet, for some people, availing of exercise, but for others it might just be a drug, because they're having to work 2 jobs, or that have too many kids to be taking time out to go to the gym, or to be going running, and it might be easier to take a pill or have an injection that ends up with the same positive benefit on insulin signaling, let's say, than having to work hard at all the lifestyle measures, because it is hard setting time aside to do exercise many times a week, and to shop well and have the very best foods is more expensive. So we have to think about the range of options to help everybody, and not just the privileged few.
And going back to epidemiology, so in the United States, and I think the UK is pretty much the same, if you look back to, say, the early 1900s, or even mid 1950s, 1960s, obesity was less common, type 2 diabetes was less common, and you're right, these diseases are affecting mostly the lower socioeconomic status that have the issues you talked about, and plus get a lot of advertising, and they can buy calorie dense food pretty inexpensively, but they're not getting them good food. But and of course we have like baby boomer generation here. There's more people, the demographics is shifting, so there's more people in the older age groups, which means there's more people that get Alzheimer's, Parkinson's. But is there any evidence that if you kind of control for that, is there any evidence that there's increased incidence of Parkinson's or Alzheimer's that's independent of just more people being in the age group where they're at risk?
So I don't know. I think I understand you that, it is the epidemiology suggestive that as the environmental risk factors, including obesity,
Right.
are more prevalent, is Parkinson's become more prevalent. I suspect if looked through for well enough, that is what we would find. The difficulty is that there are so many confounders, including our measurement of exposure, over a life course, and our measurement of Parkinson's disease, and at what point you stop measuring, because some of the generation who've been exposed to the biggest risk haven't yet reached the age of biggest vulnerability. But I suspect the cohorts that are being studied from over time.
Well, the bad news, the bad news, I guess it's good news for epidemiologists, but the bad news for people is that childhood obesity is increasing. There's a lot of kids now, even middle school, high school age, that have obesity. Even some of them are developing hypertension and type 2 diabetes. So that's the cohort that when they get to be 60, 70, then you'll have enough data to say one way or another whether that metabolic state is contributing.
Yeah, on a public health level, having funding to help support diet and exercise really from all children in school, it's desperately important, because if you set people off on the right trajectory, they're much more likely to stay on that trajectory than trying to rescue someone who's already developed, whether it's obesity or a sedentary lifestyle or poor dietary habits. What happens in childhood will tend to persist over subsequent decades. So it's worth the long-term investment to put that resource into the schools.
And unfortunately, it doesn't. The way that in profit-driven societies, right, capitalist societies, people are bombarded with information and actually worsens their health, right? Advertising, the fast food industry and so on, and even to some extent the pharmaceutical industry. People have this impression that if they get sick, there'll be some treatment for them. But in case of Alzheimer's and Parkinson's, hopefully in Parkinson's, maybe not anymore since your work, but but still, no really dramatic things that are going to reverse the disease and put you back to normal. So there has to be like some public-wide effort where you educate people and enable people. You mentioned people having to work 2 jobs or whatever, and they've got kids and they don't have time to exercise, and they're getting fast food, don't have time to make healthy food. Yeah. How does society enable them to do the things that will help them and their children?
Yeah, it just has to be a tax incentive. Things that are good for you have to be cheaper than things that are more expensive.
Yeah,
that are bad for you, and that is possible. To reduce. In the UK we have VAT, which is across the board, and it tends to be that the more healthy options are more expensive, and I'm sure it's exactly the same in the US. But
but Tom, there's here you have lobbyists in the UK.
Yeah, I mean,
So
yeah,
they for your decades since I remember, some when large scale agriculture came in, so you have monocultures, just corn, just soybeans, just wheat, right? And the government is subsidizing these big farmers. Of course, I was back at the University of Iowa in the early 80s when I was mid 80s when I was a graduate student. These big essentially companies taking over the small farms, buying their land. And so we lived in this small town outside of Iowa City, and my wife and I had just gotten married then. A very small town. It had a bank, it had one gas station, and I don't know, 300 people. And one day there was a lot of sirens going, and so we talked through word of mouth what was happening. And so some farmer had come into the bank and shot and killed the president of the bank. And so what happened was that he had a loan from the bank, and he couldn't pay the loan or something. Anyway, there was this whole thing where the small farmers were in trouble and the big farmers took over buying up. And so now it's hard. It's corn, soybeans, wheat are cheap.
yeah.
And a lot of that, healthy variety of vegetables and fruits are expensive.
I suspect you're right that it's all very well for us to say, well, healthy foods need to be made cheaper, but you can't argue with the major corporations that control all the have all the financial muscle, and all the political
Yeah.
power in their grasp as well. I suppose what it means is that relying on people's behavior, whether it's diet or exercise, is not going to be the only solution to neurodegeneration, and therefore we need to find pharmacological rescues, where people aren't able to access these things in other ways. And the GLP-1 receptor agonists will hopefully be proven to be the case, not for Parkinson's, possibly for Alzheimer's, possibly for other forms of neurodegeneration as well. And the biggest challenge that we've got is trying to make it attractive for the pharmaceutical industry to take this knowledge, which has come from the academic community, not from the pharmaceutical industry, and apply it to the neurodegeneration world. Because at the moment, all of these companies have their GLP-1 receptor agonists for diabetes, and they, in a sense, they want to protect the diabetes market that they have, or the obesity market they have, and not take a risk with the drug. If they have a negative trial, then that might undermine the diabetes market. And it is a high risk just investing in neurodegeneration research, because for so many decades it's not brought any successes.
How expensive is, I guess I don't even know that. How expensive is exenatide?
It's pretty cheap. So in the UK, it's about £100 a month. So £1,200 a year. Pound is the same as a dollar these days, pretty much. So it's not hugely expensive at all.
Yeah, that's good. Good. Okay, so I'm looking forward to see in the years to come how this progresses. There was some, I guess I did want to mention gene therapy. So there are some cases of instances of Alzheimer's disease that are caused by genetic mutations. And there's a, big literature on this in the United States. Michael J. Fox, maybe, I don't know if he's even been genotyped. Maybe you know. But I've never seen anywhere where he's been genotyped. But he was in his 30s, I think, when he first started having symptoms.
Yeah. I'm sure he's very likely to have a genetic contribution to it, but I don't know if he's been genotyped. But of course, that's the thing when we're talking about treating Parkinson's, we have to think about heterogeneity. We have to think about people. The commonest gene is the GBA1 gene. It's not Mendelian, but it's a very strong risk factor for developing Parkinson's. It's the gene that causes Gaucher's disease if you have two abnormal copies. And there's a gene therapy program, or in fact, a number of gene therapy programs looking to see if they can replace the enzyme that's missing in GBA1 associated Parkinson's disease and boost its function. And that's it's mechanistically very sensible. You're just treating the underlying cause with a therapy that should transfect the relevant neurons and engage the target, and that should be successful. And likewise, there's LRRK2, which is a Mendelian cause of dominantly inherited Parkinson's disease. Not quite as common, but commoner in Jewish and Mediterranean populations. And in that group, you can use a LRRK2 inhibitor because it's a toxic gain of function. And Denali and Roche and Roche or Biogen, one of the two, have partnered to make a LRRK2 inhibitor, which is orally active, CNS penetrant, and hopefully that again is by engaging with the upstream cause of neurodegeneration will make a difference.
And the LRRK2, that's dominantly inherited?
Yeah.
I've always thought, if I had, like, if I had family history of inherited Alzheimer's, Parkinson's, and then I got genotyped and I found I had the mutant gene. So I knew I had the 100% chance of developing this if, I live in the 50s or 60s. That I think this is something that children should know. In other words, if you have a parent that has a mutant gene that causes Alzheimer's or Parkinson's or any other disorder, I think I would want to know, because then I could decide, do I want to have kids? Well, first I'd genotype myself, right? If I'm positive, then I'd say, well, I don't want to have a kid that has this. This is really bad. So could adopt, or you can there's actually a way you can ensure that your child doesn't have the mutation. That's in vitro fertilization and embryo selection, which is a little expensive. So in theory, these dominantly inherited disease could be eliminated in one generation, right? If you had education, of course, everyone would. What are you thinking on that?
Well, I mean, this is an ethical and a moral, argument. Some people who have a LRRK2 mutation, may never develop Parkinson's, or they may develop Parkinson's and have mild symptoms, and they may feel that they've had a very rich, rewarding life despite getting Parkinson's disease, and that they wouldn't want their genetic status to have any influence on their family decisions, and so everyone will have their own views about that, and you can take this argument. We can learn a lot from the Huntington's community. Of course, it's far more highly penetrant at a lower age. So I think we have to think about finding for these Mendelian genes. You want to find treatments which should be more easy as we are getting better at gene delivery, gene editing, all the viral vectors. There's lots of ways of delivering genetic information to adults, whether it's oral, intravenous, stereotactic.
And there are the brain's a little harder than. So there are gene therapies now for certain blood cell disorders, and some eye disorder. I can't remember what it was. They actually inject
Yeah, for
into the eye,
maximization.
the
Yeah.
construct, and do gene editing. So yeah, I think this is on the horizon. A lot of work going on in that area.
Yeah. So the GBA1 they inject into the cisterna magna, so it can access the CNS rather without having to worry about the blood-brain barrier. And of course, you can do stereotactic injections into the putamen. That there's still interest in using growth factors and using a gene therapy as a way of having chronic administration locally of a growth factor.
So you use a viral vector to express the growth factor?
Yes. Yeah.
Yeah.
That's still work in progress. Yeah.
Yeah. Okay. Well, we've been on for about an hour, and I hope the viewers and listeners enjoyed this, and then they're excited that there's potential treatment for Parkinson's. We'll see how the phase 3 trials, that'll probably be a few years, right? Before.
Well, the last patient will finish next year. So we should be looking to publish by the summer of next year.
Were you delayed by COVID, I assume?
To some extent, but we couldn't recruit during our lockdowns, but we still managed to follow up the people who had already been recruited, and so we survived where many others just had to pick up and go home. So
here they shut down a lot of things. We had a trial of intermittent fasting in patients at risk for Alzheimer's. Demetrios was heading it. It's done now. We're starting to write it up, but it was people with obesity and insulin resistance between the ages of 55 and 70. So they were randomly assigned to intermittent fasting or control, which is advice for healthy eating. And then it was, 1 year study. So kind of like you did at baseline, and then at the end, cognitive testing, functional MRI. Do you do MRI on your patients?
We're not in the clinical trial. No, we're doing a scan in a subset.
Functional MRI, took blood, CSF, and then Demetrios' group was doing a lot of extracellular vesicle stuff, kind of like you did. We're looking at some other things in addition. And so there are some interesting results, so stay tuned for that.
Excellent.
Okay, Tom.
It's been lovely talking to you.
Yeah, I enjoyed it a lot.
Okay.
Cheers.
Cheers. Bye now.
Bye.